other functions of media. So here we come to the idea of media being useful for diagnostic purposes, right? Two types of media that can help you to identify microbes. One is selective. So when media is selective, it means that it's like a college. College admission is selective, more or less selective, right? What does that mean? Huh? Not everybody gets in, right? Like Lakeland is not technically selective. Okay? If you can read and write, multiply 2 by 2, which is great. In my personal opinion, it's great. Because it gives people, seriously, you can start from any level. You can start from high level, from low level, but that's just digression. So, like TSA is not a selective media. Anything can grow on it. Well, almost anything. Selective media will allow a very specific group of microbes, or sometimes even the microbe, to grow. Okay? So it selects. You can see you have a wide variety here and only certain type on the selective media. How? Usually selective media contain something that inhibits growth of certain microbial species. One of the selective media that we're going to work with is mannitol salt agar, MSA. It contains two chemicals, as you can say from the name. One is mannitol, and another one is quite literally salt, sodium chloride, right here. And concentration of sodium chloride in MSA is 7.5%. Percentages are usually kind of sort of irrelevant. So let me give you an idea. 7.5% means that in 100 milliliter of a solution, there's 7.5 grams of salt. Is that a lot? Imagine that you put 1 tablespoon of salt per cup. Is that salty? God damn, it is salty. You won't be able to drink it. You will have troubles dissolving it. It's not easy to dissolve, okay? So, not most of the microbes cannot survive on that high concentration of salt. Staphylococci can. So it will restrict, and we will see that. We will be able to specifically show that while staph can grow, strep cannot. Okay? And a lot of other gram negatives cannot and stuff like that, okay? So it is selective because of the salt. What about mannitol? Mannitol is the sugar. What is the main function of sugars? Energy. Yeah, food source, energy. Now, Staphylococcus can ferment sugar. Which products of fermentation do you know? Okay, carbon dioxide will be a product of alcoholic fermentation, but unfortunately, stuff doesn't produce alcohol. Something else. That's alcohol. I would love if stuff would produce ethanol. If you have stuff in your nose, you would be buzzed all the time. Something else. What are also fermented products that you can buy in the grocery store? Huh? Yeast is something that ferments. Say, yogurt and cheese. What about pH of a yogurt? Acidic, it's acid. They produce lactic acid. It's a lactic fermentation. Huh? Question? Lactate, yes. Lactic acid, lactate. So lactic acid, it's acid. What kind of pH? Lower than 7. This agar has phenol red as an indicator. When pH drops, phenol red turns the color to yellow. If these species of Staphylococcus, like Staph aureus, can ferment mannitol, colonies will turn yellow. Make sense? colonies or even area around them. If species of staph, like Staph epidermidis, cannot ferment mannitol, they will remain red. Does that make sense? So now it restricts growth to a certain species, a certain genera, or genus in this case, the Phylococcus. Moreover, this particular medium allows you to distinguish different species in that genus. And we'll see how this ability is called. These are some examples of selective media. I want to highlight few that we may work with or just of interest. Tomato juice agar. What is the pH of tomato juice? Acidic. So this agar should be highly acidic. It's used to isolate lactobacilli. Lactobacilli are microbes that actually can make different dairy products. Like not cheese, it's different stuff, but yogurt or buttermilk. Okay? Acidic environment is normal for them. So that highly acidic environment inhibits growth of everything else. McConkey agar. It's a to-go choice in growing gram-negative microbes from intestine, enterics, and it has bile. Bile by itself, bile salts, are protective. They inhibit growth of gram-positives, but allow growth of gram-negatives. Does that make sense? See the difference? Saburot agar that we're going to use next week. It's acidic with pH about 5 agar that inhibits bacterial but supports fungal growth. One question that out of 3 classes only one person asked. Do we have to know all that stuff? No. You don't have to memorize this table. God forbid. Okay, because if you don't use it, you lose it. You must understand the concept. Does that make sense? So, not only the theoretical phrases like this, but be able to distinguish based on like a real life example. I will tell you, say, tellurite medium contains inhibitory potassium tellurite, and only species that can grow on it is Corynebacterium diphtheria. How would you call this medium? And you have choices. And you pick selective. Does that make sense? So you have to be able to name the actual example. Any questions on that? Differential. Remember we just talked about MSA, mannitol salt agar? It allows different species of Staphylococcus to grow, but also helps you to differentiate them species. So differential media supports the growth of multiple microbial species or genera so that those species or genera show up in different colors. Make sense? So here, general purpose, they're all the same. Here, they appear in different colors. Mannitol salt agar is both selective, because it selects for gram-positive staphylococci, and differential, because it helps you to differentiate between mannitol fermenting and mannitol non-fermenting. Make sense? On the last slide, I mentioned McConkey. If McConkey agar contains lactose, microbes that can ferment lactose and produce lactate and drop the pH will change the color of the indicator, the neutral red. Now, McConkey agar is both selective and differential. it supports the growth of gram-negative microbes from intestines and allows you to differentiate. So, okay, let's put it in a better. It selects for gram-negative microbes from the intestines and differentiates those gram-negative microbes based on their ability to ferment lactose. Not so straightforward question. If you have 2 gram-negatives on McConkey agar and one can metabolize lactose and one cannot, which one would you think be more likely pathogenic and which one will be more likely your normal commensal microbe? Will be, you think? It will flourish on the lactose, and the one that doesn't will be okay. In fact, it's opposite. Reason? We eat lactose. So if that would be true, every time we drink milk, we would have massive bacterial infection and die. Which, as far as I can understand, people with lactose intolerance, that's pretty much what happens to them. Well, quite close. They don't die, but very unpleasant. But lactose intolerance comes from a different thing. It's not about microbes, it's about your own gene expression. Now, lactose is a component of human diet. From very first days of life, actually. Okay? It's a component of human diet. Microbes that are commensal in a gut, they co-evolved with humans and can use lactose as their food source. Invaders, usually the ones that cannot. That makes sense? Like if somebody claims that he's Russian but doesn't drink vodka, that's not Russian, right? So you can subject them for a test. As you know, Russians drink vodka from like the age of 2. So they evolved to do that. So that's another example of differential and selective media. There are others. We're going to use this and this. We're also going to use Blot agar. Blot agar, remember we talked quite a few slides before about Blot agar as being enriched by blood. So it is enriched and at the same time differential. It allows you to distinguish microbes that can cause hemolysis versus ones that cannot. Does that make sense? It's not selective, but it is differential. Okay? This, bless you, these are 2 real-life examples. This and this of differential media. First is triple sugar iron agar. So it contains 3 different carbohydrates, glucose, mannose, and I think arabinose, and iron. So first one is no microbe. That's how it's supposed to look. Second is microbial growth. Does anything happen in the second one? In terms of the color or anything? Not really, no. Not really. I mean, it grows, but does nothing. So it means it does not ferment sugars, and it has nothing to do with iron. Third one, what do you see? Change of color. Where? Bottom. What's so weird about the top? Why there is no color change in the upper portion? Why? Huh? Oh no. It moved all the way, but for some reason it cannot climb to the top. Why? What's different between the top and the rest of the tube? Yes, oxygen. So it's anaerobic bacteria, yes. It is not only anaerobic, it's anaerobic that is obligate anaerobic. It cannot tolerate oxygen. Okay? Does that make sense? So it cannot grow here where oxygen is present. Does that make sense? So now look, gives you fermenter, anaerobic. This one will be different microbe. What does that mean? It can mean many things. It's either facultative anaerobe, so it's a microbe that may use oxygen but don't have to. So if it grows up top, it uses. If it grows at the bottom, it does not. But most likely, it's aerotolerant anaerobe. We'll talk about it. It means that it doesn't use oxygen, but it's not getting killed by oxygen. And here, you can see the black layer, okay? That's, again, sulfur-reducing microbe. The one that reduces sulfur, and it forms iron sulfide. Iron sulfide precipitates, gives black color, and you can say that this reaction, the sulfur reduction, happens in the presence of certain amount of oxygen because it's in the upper layer, not in the bottom layer. So you see, some combinations of reagents can provide you with a ton of information about the microbe. This is something. Now, many times, okay, those pictures were taken from outdated microbiology book, which was published like 9 years ago. And trust me, every microbiology book that is published is pretty much outdated at the time it is published, because the field develops so fast. And you may say, oh, come on, this thing cannot be modern. Well, that chromagar is advertised in pretty much all microbiology scientific magazines. It's a major tool to diagnose urinary tract infections. So on that plate, you can see the names of the pathogens. E. coli, Klebsiella, Proteus, Enterococcus faecalis, Staph saprophyticus, Staph aureus. Those are most common pathogens of urinary tract. And each of them, colonies of every one, has a specific particular color. So see, E. coli is like dark pink, Enterococcus is light blue, and so on. So you can take the sample from a patient in which you suspect the urinary tract infection, put it on the plate, and you see what they have. Does that make sense? It is really like a modern thing. Any questions on differential selective? Not really. Viruses. They gotta have something to grow. What it is? Cell. They gotta have cells. So technically speaking, media for viruses are cells. And cells also have to be grown in the medium. So this is kind of just a list of components of the typical medium for cell growth. This is how cells are grown in the lab. You have a plastic flask. Cells grow on the surface, and they are covered with a thin layer of growth medium. So when you want to infect them, you add virus, and you get your infection. That make sense? But you get the cells. But you get the cells. Narrow it down. Which part of an animal? You can get from blood. Can you get from liver? Sure, why not? Like virocells, they are cells of the kidneys of African green monkeys. Nothing funny. Those poor fellas, they go through quite a torture. Well, I mean, they anesthetize, but anyway. So you can get from any tissue. You can get cells from any tissue. You can get them from blood. I donated my own blood for research purposes for my colleagues. They needed my white blood cells. You can get them from liver, kidney. You, at least theoretically, familiar with circumcision. Those flaps of skin. What are they used for? Well, guess what? Cells, human foreskin fibroblasts, are prepared from those skin flaps. And you can buy them. I mean, not the flaps, the cells. Okay? You can buy those cells. There's it's not a company, it's non-government, non-profit organization that it's like a bank, a biological samples bank. You can buy all kinds of cells from those that bank. Have you heard about a lady named Henrietta Lacks? You have. What's so special about Henrietta Lacks? Say again? Yes. Yes. So, cancer cells utilized for a bunch of studies. I would say that you gave a perfect explanation. The only thing utilized for a bunch of studies is a little bit of understatement. I would say about a third of all biological research in 20th and 21st century was done on the cells that were derived from the cervical tumor of Henrietta Lacks. Okay, so I think it was like mid-50s, cell culture was just invented. She had cervical tumor was excised and used to prepare the cell culture, and this cell culture then spread through the scientific world, and so-called HeLa cells, Henrietta Lacks, HeLa. And these cells are pretty much everywhere. You walk, I would guarantee you go to Case, 3, 4, 5 labs will have them in a freezer. Cleveland Clinic, CSU, they're all going to have HeLa cells. There's a little bit of a problem with that story. If you know, don't say. What was the problem with cells derived from cervical tumor of Henrietta Lacks? Doesn't have anything with biology. Yes, no consent. Now, why people who didn't get her consent didn't get persecuted? Hint, it's not because they didn't get caught. If you don't get persecuted for something, what does that mean? It is? Yeah, it was legal, absolutely. You didn't have to get the consent at the time. Nobody thought about it. Tumor? Consent? You gotta be kidding me. Right? Why is that? Why was that? Why it has become a problem eventually? Any ideas from ethical standpoint? I mean, not getting consent is kind of a bad thing, just overall, but why it's not? Okay, so cells are everywhere. And what can we do now in terms of genes, huh? Oh, the problem is the genie is out of the bottle. I mean, you have thousands of researchers who use them. You cannot possibly ban them. But what can we do with the genome? Not only we can recognize the sequence, okay? And if we know the sequence, it's probably the most intimate information that you can have about the person, right? Well, Henrietta Lacks passed away shortly after the surgery because of the cancer. But somebody was left behind. Her family. So, knowing her genetic makeup, pretty much know a lot about her family. And understandably, it made them very uncomfortable. Because your genetic information is the something that one particular type of business would be very much after. What type of business? What do you think? Who would want to know your genome? It's legal. It's not like drug dealers or something. Not so much. I mean, we're not at the stage where we can clone. Yes. Who? That? Huh? People who? These guys that I'm talking about, they're more powerful than sperm banks. More powerful. Oh, come on. No, it's private business. Insurance company. Health insurance. Because if they know that you have a gene that predisposes you to diabetes, they can drop your coverage, right? That kind of sucks. And about 2 weeks ago, the company named Illumina, they announced that they're going to release the machine on the market that will make sequencing of a human genome as cheap as $100 per genome. Pretty much, you have a baby, you walk in the doctor takes a blood sample, runs the machine, and you get the full genome of your baby. Like, full sequence. And you can say that your baby, I don't know, had peanut allergies or some immune disorders after like a week of life. It's pretty awesome. The only problem is who you share that information with. So that ethical problem, I just want you to be aware of that. We get consent as, you're going to get consent as medical professionals, not because of some crazy lawmakers in Washington, but because it does make sense. In some cases, there is no consent if it's completely anonymous. I listened to an interview with a virologist. She needed placental tissue for study. And where to get placental tissue? You call OBGYN. So she called OBGYN and asked, which forms should I fill out and stuff? And he said, bring a bucket. That's it. It gets thrown away. Placenta, after the birth, after delivery, it gets thrown away. So it's not under consent, and it's completely anonymous. You cannot possibly identify and link it. Okay, so that's a different story. But consent is taken because of potential ethical implications. Does that make sense? Live organisms. In some case, use plants. This is the leaf of tobacco infected with tobacco mosaic virus, and on the left is the leaf without infection. You may think about plant viruses and plant infections as being kind of inferior. Let me tell you something. I worked in University of Nebraska, and we worked with hemorrhagic fever, studying hemorrhagic fever, and we lost our funding. People who worked on plant pathogens were laughing at us. They got money from USDA and from a bunch of companies that make different kinds of pesticides and herbicides. So it's really a well-funded research, and it's important because it's a food source. Okay, you can use animals, like this image of a cute little mouse with influenza virus inside of its lungs. In some cases, virus or microbe or something else does not replicate, does not reproduce in anything except animals. You cannot find suitable cell model, or media does not support the growth, or if you need to reproduce worms, the nematodes, some nematodes, you use rodents to grow nematodes in them. In some cases, there's not even animal model. Have you heard about norovirus? Do you know where it was isolated first time? You should be proud of it. Which is right 60 miles away from here. Be proud of it. Norwalk, Ohio. The origin of two-bucket disease. Why two buckets? Yeah, you got to comment from both ends. Yeah, you shit and you vomit. That's right. So that's two-bucket disease. Now, for a long time, there was no animal model for norovirus. It was only human norovirus. Who would you use to study it? It's hard to catch. It's kind of a pain in the butt to run after people who have diarrhea and vomit and then check if it was actually norovirus. Who would you use for studying if it's only human? Humans? Yeah. Is it going to kill you? Not really. I mean, you're going to feel very uncomfortable for 2 days. Which specific, let's say, group of humans, occupation-wise? Any ideas? One of the classes, they gave it right off the bat. No. Well, I mean, we're dedicated, but we're not that dedicated. Huh? Yes. College students. $300. So on Friday you drink. It's a fecal suspension. You drink on Friday. You spend weekend in the toilet. Well, in the lab actually, because they collect samples. And on Monday you back to school. It's pretty exciting. I know that in some cases medical students did it for extra credit. Huh? Yeah. Now you can use humans, okay, but the animals or animal product or animal live media that we use most frequently are eggs. You can see that lady injecting influenza virus in eggs. We produce influenza virus for vaccine in eggs, in chicken eggs. So what are the disadvantages of eggs? Do you have eggs at home? Yeah, you have to handle them with caution. Something else. Well, it's more of an advantage, I would say, because you can tweak them. Okay, but I think I know what you're talking about. They can be different. So these eggs have to be free of pathogens. So that makes them more expensive. What about each egg is a cell? What about the size? Big, okay? They occupy a lot of space. Probably well, why did we use them then if they saw disadvantages? Animals were more expensive and we didn't have cells in the first place. But the biggest disadvantage of eggs, what makes eggs? Chickens. Chickens alive or they are soulless machines? Yeah, but some may argue otherwise. But anyway, if they are alive, what they can do? Any live creature can die. Well, they can reproduce, and I believe they do it, but they can die also. A couple of years ago, the egg prices in states went up because chickens were dying, really, and it actually affected the vaccine production, the flu vaccine production. There was not enough special pathogen-free eggs for making vaccine against flu. You know what was killing chicken? Chicken flu. I think it's quite ironic that chickens were dying of the flu, didn't produce eggs, so humans couldn't get enough vaccine against their own flu. So we now try to move from, well, not we, but some of the producers of flu vaccine try to move from eggs to cells. Not all of them. What do you think can be a problem with moving from egg to cell? What is the main obstacle? Why vaccine producers don't switch to cells? It has nothing to do with biology. It's much cheaper. Cells are much cheaper. Not really, no. It's probably going to be much faster as well. You have tons of banks, so cells, by all means, are much easier to handle. It's not about technique, it's not about biology, it's about laws. When you change something in the makeup, you have to test, and you have to get permissions. And it's hundreds of millions of dollars in years of work. Nobody wants to do that. So once you inoculate it, you have to incubate and isolate your microbes. If you really want, I can show you incubators, or you can pick at them through the window. Nothing interesting. It's, imagine the fridge, but not a fridge. So instead of +4, it's +37. That's it. That's the whole difference. Okay, and it's dark inside. Now, why incubator? Why not just leave the plates right over here? Controlled environment. Why controlled environment? Why it's so important? Okay, if microbes grow only at a certain temperatures, but some of them will happily grow at the room temperature. Still, why controlled environment? Cross-contamination, I can put the box, like, and seal it right here. Skewed results, somebody else was going to say, Elliot? So, skewed, not necessarily skewed, but what is this? Say it? Temperature here, and temperature in incubator. In incubator, it's always 37 degrees Celsius. What about here? Fluctuates, right? So, we don't really know what was going on. So if we want to replicate the experiment, we can't. Does that make sense? Because we need to eliminate the variables. If we study something, we have to study one variable. Make sense? Also, if somebody else wants to study it, we can't just say grow at the room temperature. Because their room temperature may be very different from our room temperature. Right? So controlled environment. Also, we can control for atmospheric gases. We can add CO2, add nitrogen, do whatever. Right? Humidity, stuff like that. Usually they grow in a couple of days. So we inoculated on Monday. They're ready on Wednesday. Actually, they were ready yesterday. Usually one day. But, sorry, those are fast-growing microbes. Some slow-growing microbes, like Mycobacterium, can take days and sometimes weeks to generate first colonies. I wouldn't be able to work with them. I'm too impatient. And then, you've got to isolate. Those samples from hand wash that you had. Can you open it, please? Thank you. Beware, it can lock you out, because the constantly open door, it shuts down after a certain period of time. So when I open the door, eventually it will become locked. Isolation. Why do we need to isolate? And actually, so if we want to grow microbes in a pure culture, we have to pick a microbe from the colony, right? Pick a little bit. And either put it on another plate or directly in the liquid medium. Liquid medium is quite obvious. You have white colony, you pick it, you throw it in the medium, and Staph epidermidis grows in the liquid medium. Make sense? What is the reason? Why would you want to do another plate? Why do you want to subculture it once again? Yes. Excellent. If the colony that you pick, if you're not sure about it, okay, so you pick it, but maybe it appears white, but maybe it's contaminated. So you want to spread it and see if it's contaminated or not. Does that make sense? So how we separate those colonies? So here you have mixed sample, and then you somehow spread this sample and see separate cells. And this is an example of mixed sample. When you do some magic to it, colonies become separated, and you can see now distinctively 3 species: Micrococcus, E. coli, and Serratia. Here, there's no way you can sometimes even see them. So how do you do that? this you use the streak plate. And I don't want to play with that thing, the computer a lot. So I will try to make a picture so it's going to be a part of the video, how you do the streak plate. Essentially, the idea of streaking, the idea of separating species is to dilute the sample. Actually, you can dilute your sample right on the plate. Here how you do it. This is your plate. Okay? You divide it like this. You can actually, we're gonna do it, you can draw it with a Sharpie. And then you have your initial inoculum. Put it right here. It's gonna be thick, dense, a lot of microbes. Then you sterilize your loop, okay? And you do this. You pick a little bit from here, and with this, inoculate this part. Does that make sense? So you just picked one little bit and moved it to another quarter of the plate. Since it may not thin it out enough, you pick a little bit of this and move it here. Does that make sense? You subsequently thin out your sample. Are we clear? Good. This is a very important technique. Again, it's a cornerstone of proper lab technique, knowing that streak plate, how to thin out. Does that make sense? Huh? We didn't thin it out. So we did streak plate. We did a simple one. So what you essentially did, you took a plate and just inoculated it like this, right? Well, half of it. That make sense? The next time when we're going to collect the environment samples, this is what we're going to do. So you will swap something, okay? And probably, since we want to sample as much as we can from the environment, it's really fun. What we're going to do, we're going to take the plate, split it in quarters, and then you're going to inoculate one sample here and thin it out here. So at least you can separate them, okay? Because if you swap something like your wristband, there's going to be a lot of microbes. They're going to be boring, but there's going to be a lot of microbes. That makes sense? So, and second half is going to be a different one, right? So that's most common method, the streak plate. Now, other methods to spread your microbes are pour plate and spread plate. In pour plate, you inoculate liquid agar, the worm. Warm enough to stay liquid, but not too hot, otherwise microbes will die. So inoculate the agar, mix it up, re-sterilize the loop, pick up a little bit from here, move it to another tube with agar. So you dilute your sample. Does that make sense? And then re-sterilize the loop and move to next tube. You can do as many dilutions as you want. Eventually you will get less and less microbes in each subsequent tube, and they will be thin enough. So this is an example of, it's a result of that, such thinning. Another way is the spread plate. Spread plates don't thin it out really. You put the sample in the center of the plate, you take a hockey stick, and you just spread it on the plate. It's very useful when you want to make so-called bacterial lawn, when plate is all covered. Okay? Inspection and identification. Well, inspection, we're not going to have a quiz today, no worries. Inspection. You did that already. Often, when you know the source of an inoculum, and you know, and you see the colonies, the size, the shape, the color, you pretty much, you can guess what it is. Not guess, but pretty, identify them, almost. So, skin, Staph epidermidis, Micrococcus luteus, we already know that those are two major types. You look at them under the microscope, definitely cocci. We could do some gram staining, see that they are gram positives. Those huge big ones, we suggested those were bacilli. We could do gram staining on them as well. They should have shown up as gram positive as well. Bacillus is gram positive bacterium. So we can do a lot with at the inspection stage. We can describe. That's what I would really like to see in the results section of your lab reports. Brief description of what you observed. Colony size, colony shape, colony color. Okay? Number. Did you have a lot? Did you have just a few? Okay. And then you do identification. That's the part when you figure out exactly what it is. There are three major approaches. Immunological approach. Are you familiar with the concept of antibodies? I'm going to ask a question different. Do you want me to give you a quick wrap about antibodies and what they are? Okay, I will. It's going to be very quick, very basic, okay? We'll get immunological, biochemical, metabolomics, fancy word. You can identify a bunch of metabolites of the microbe, what it produces. It helps you to identify them, okay? Genetic molecular biology. You can identify microbe based on the DNA sequence, okay? We're going to chat about each of these methods separately, and this is something that's usually not well described in textbooks. I don't know why. Maybe authors think it's too complicated. My personal opinion is you don't have to know the method or technicalities or how the machine works, like all the details and stuff. But I want you to have a basic understanding of what's going on. Because when you walk into that hospital and you talk to people from clinical lab, they will use words that you're going to learn about today. Words like immunostaining, immunofluorescence, PCR. I don't want you to look at them stunned and think, what the fuck are they talking about? And I don't want you to pretend that you are an expert. It's better to say, look, I've never done it, but I understand these words. I know what you are saying. It sounds familiar. I understand that, you're looking for DNA fragments or for antigens, right? So first methods that we're going to chat about are immunological methods. They're all based on one profound feature. If you put something foreign in the animal, like mouse, something that's not mouse, something that we call antigen, mouse will produce antibodies to the antigen. Antibodies are extremely specific. Antibodies to a specific protein will not recognize any other protein, only this one. They are fantastically specific. Make sense? This whole interaction, specific recognition of antigen by antibodies, is what is as a foundation of immunological methods. First one is Western blot. I'll get to the name. I'll explain you why it's Western. This method allows you to detect specific proteins of a microorganism. Of course, you have to just kind of disrupt the microorganism first, right? And then you can make this sample of all proteins of a bacteria and put it on the gel. Apply electricity to it. Proteins are charged, so they're going to go from negative to positive charge. Does that make sense? Because they are charged, they're negatively charged. Which ones, smaller or larger proteins, will go faster? Smaller. Smaller will go faster. So smaller will be on the bottom of the gel, larger will be on the top. Gel is awfully inconvenient to work with. Seriously, it's like this jello looking thing. What you do next is you take the gel and take a special membrane, a material that is like it looks like a paper, and you put gel and paper next to each other. You apply electricity again, and proteins go from the gel to that membrane. That makes sense? Now proteins are in the membrane, it's easy to handle, and you expose membrane to the antibodies. Antibodies against the protein you are looking for. So say I want to know if my sample has M protein of Streptococcus. I take antibodies for M protein, I take sample of streptococcal lysate, do all this stuff, expose the membrane to the antibodies, and if there is a protein, I'm going to see a band. Antibodies will bind to the protein and they will either light up or do something other. Some other things. That makes sense? So identify a protein, and you identify them because you can separate them. Does that make sense to you? Or not really? Now, this is something I want you to understand. When I ask, does it make sense? It's not a rhetorical question. You are absolutely welcome to say, no, that does not make any freaking sense. Can you explain it again? So if something isn't clear, please let me know. I cannot read your minds. Okay, so we have antibodies that recognize a specific protein on the membrane after the separation. Pretty much the same. As long as you get the idea of antigen and antibody, everything else is the same, just slightly different technical arrangement. Eliza. You have the antigen that sticks to a surface, and you have antibody that recognizes it. If there is an antigen on a surface, there's going to be light, for example, or change of color, because this antibody is labeled in a special way. If there is no antigen, there's no change of color. The most common, though, is this type, the sandwich ELISA. When you have, and ELISA stands for enzyme-linked immune sorption assay. You have antibody that will capture the antigen. You add another antibody, well, two of them, that will detect the antigen. Does that make sense? So if you want to do that, most of the companies will sell something like that for detection of, I don't know, influenza antigen or HIV. HIV test is based on ELISA. You can buy plates. They have 96 wells, and you can put samples in each of those 96 wells. And it's, yes? So, unless there is a complex, okay, if there is no complex, there's going to be no signal. But if there is a complex, there's going to be signal. Did I answer your question? Okay. Sometimes immunofluorescence is used. Immunofluorescence is great if you have like a tissue sample, a blood sample. So you don't even have to culture. Just have a sample, you add antibody, they stick to the antigen on the surface of the cell, and they light up. You can have different colors. Like here, that's the courtesy of my good friend from Houston. So red ones are neurons, green ones are astrocytes, and blue ones are nuclei. So you can see 3 different structures on the same image at the same time. I think it's awesome. You can diagnose like 2 or 3 different microbes in the same sample. It's pretty neat. Biochemical. I'm going to talk about 2. They're probably more. Chromatography and mass spectrometry. They're not essentially chemical. They are physical, I would say. Chromatography. I don't know if you've taken analytical chemistry, but you can actually do chromatography at home. I'm serious. You can at least try. You can take several dyes, like, you know, the acrylic paint or something that's preferably water soluble. Okay, so watercolors would be best. You mix watercolors together and you take construction paper, white construction paper, and make a strip. You put it in those watercolors and you wait. What's going to happen to that solution and the paper? Huh? Yeah, it moves up the paper. Capillary forces, right? The dyes will move together with it. Depending on the structure of the dye, some will move faster, some will move slower. And at a certain point, you may be able to see lines of different colors. Does that make sense? So you separate your dyes. It's not preparatory, it's analytical mostly, but you can actually, sometimes you can even use it for prep. So this is how it works. You have a column in this case with some kind of a filtration material, a sorbent, okay? And you put your sample in that column. And different fractions of the sample move with a different rate. They can either move because of the gravity, or you can apply pressure, okay? And then they will come out at a different time. Does that make sense? No? No? You know what sumo, right? The sumo fighters. Starting line, 100 meters. You put Usain Bolt. You know who Usain Bolt is? The runner? The sprinter? Okay, you put a sprinter, like a professional sprinter, professional athlete. Okay, it's a football season. You put Adrian Peterson, me, and a sumo wrestler at the starting line. We are all in the same group. And go. Who's going to finish first? Adrian Peterson, right? Then, if I'm any good, it's going to be me. Although, I would say you can bet on sumo wrestler. If he runs over me, then I have no chances. But anyway, you have separation based on a quality, a speed of running, right? In this case, you separate them based on something else. How fast they go through that filter. Make sense? Some of them will go faster, some of them will go slower. It may be based on the size, the charge, something else. Does that make sense? And then you can either collect fractions and use them, or you can put some sort of a detector here, and it will picture you like little peaks. You can see what's going on there. And you know that like this compound, it takes 5 minutes to go through. This compound, it takes 10 minutes. So you can detect. That's very common. Chromatography is very common. Another thing is mass spectrometry. This is something that technically is horribly complicated. Conceptually is simple. You have a molecule like a protein and you break it in pieces. Pieces are going to be of different size. Do you agree with me? And they also are going to have slightly different charges. So each piece has unique size and unique charge. Make sense? And then you kick them, you accelerate those pieces. That makes sense? Which ones are going to fly farther? Smaller, lighter ones. So you can separate them based on the mass. And if you put them, those flying pieces, between two magnets, depending on charge, their trajectory is going to get curved. And depending on charge, it's going to get curved to a different extent. Does that make sense? So you separate pieces by mass and by charge. Now, method is designed so that every time you break down a molecule, you break down it in reproducible way. It's always the same. Does that make sense? Imagine that every time you break the glass, it is broken in the same pieces. Like same glass, same pieces. Does that make sense? How it is applicable to microbe identification. You can do it to the whole microbial cell. You can take a cell, break it in pieces, stick it into this machine, turn it on, and it will give you a graph like that. That will show you how many of each piece you have and where are they located based on their mass. By itself, it's not informative. But if you know this picture for, let's say, E. coli, you already know, you have a reference. You take your sample of a bacteria, you run it through mass spec, and you see exactly the same picture. It's E. coli. The whole thing, the whole run through and computer software analysis of the picture, 2 hours. Extremely quick. And you pretty much, it's very simple, very quick. What should be a downside if it's simple and fast? Yeah, $750,000 for the machine. Not every hospital can afford it. Like the biggest hospital can have it. And you also have to have a culture, pure culture, something. Does that make sense? That's pretty crazy. We're going to stop. We're going to chat about some genetic methods on Monday. I hope you're going to have a great weekend. Stay warm.